Great. Thank you, Andrew. Thanks for getting us set up here. And again, welcome everybody to the presentation by Biomea Fusion, the CEO, Thomas Butler. My name is Hartaj Singh. I'm one of the biotech and technology analysts here at Oppenheimer. This is the morning of our second day, and real pleasure to have Tom present. I'm going to have to tease Tom a little bit here because I actually knew Tom when he was this big, professionally speaking, and now he's a CEO and chairman of a company that's doing some pretty cool stuff. So looking forward to this presentation. A short intro by Tom, and then we'll get into the fireside chat. So thank you, Tom, for being here and presenting.
Yeah, thank you for having us, Hartaj, and good morning, everybody. 2021 was an incredible year for the company as we really grew up in a big way. We kicked off the year by writing our S1, going public in April, raising about 175 million, all while having about 12 to 13 employees, but an incredible molecule called BMF-219. And really what we did was be able to raise enough money so that we could properly explore this covalent irreversible inhibitor of menin. And we wanted to make sure that we didn't only explore it in acute leukemia, but explore it in other liquid and solid tumors. And you really need to have the capital to do that and the internal capabilities to do that. So that meant throughout 2021 was building up the team, getting the right people on board, and then also executing on our platform because we know that for a company to have long-term success, you have to have a very deep and rich pipeline. And so we wanted to make sure that we also raise enough capital to get the innovation center built here in San Carlos. Again, continue to hire key R&D folks to continue executing on our platform, which is to build novel irreversible covalent inhibitors, and then continue that execution phase with BMF-219. And going through the summer months, writing a 5,700-page IND, getting it cleared on time or early by the FDA was huge for us. And then going through and making sure that we get the right sites, get the right investigators excited about what we're doing. We're doing something very, very different, very unique in how we target menin. And that's why we think that we're going to have great success beyond acute leukemia as well, just because of how unique BMF-219 hits menin. And then again, with the build out of the lab and executing on our second and third program, we're on track to announce our second program, in the next 3 months or within the next quarter, to announce our second program. And again, picking targets that are clinically validated. We're not taking that clinical risk, not knowing if you shut down this target, will you get response? We know you will because it's already been studied. And then we just want to make sure we get the best molecule into patients based on this mechanism of action. And so we're very excited to announce to the world this second program. And then continuing to hire, which wasn't easy in this tight labor market, as you all know. We went from about 12 or 13 employees to now we're over 60. And we take our time to hire the right people. We were very fortunate to be able to leverage our internal network. I would say 90+% of the new hires were based on word of mouth or internal network. And that just speaks volumes, right? Because then you get to hire people that you have experience with, and that you know exactly what they can handle and what they can't handle, and put them in the right position to be successful. And so, we have the capital, we have the people, and we have the molecule, and now it's just time to execute.
Yeah, great, Tom. Thank you so much for that intro. And like I said, it's just so fascinating. I've been doing this long enough now to see the progression of technology. To see the progression of the financial markets, and I'm talking all here about biotechnology, specifically to biotechnology, but also to see the progression of people. We have met Yumi, Ramses, other members of the old pharmaceutics teams before then and now. While you've been looking at different companies when you were still an investor. And then roughly around 4 years ago, you decided to form this company, and in a pretty short time span, have now brought BMF-219 to the market. I remind people that Biomea, you got BMF-219, sorry, not to market, into the clinic within about 3 to 4 years. On average, it takes about 6 years to go from preclinic to clinic. So can you just maybe let's take a step back, and before getting too detailed, talk a little bit about how you approached this problem of coming up with finding a disease that you were interested in, a molecule that you thought could be really interesting, and then how that broadened into a platform approach for you that finally became Biomea Fusion.
Yeah, so we were in Palo Alto in a small office on Emerson Street, and we got a knock on the door one summer afternoon in 2017, and it was this computational chemistry company called A2A, and they had this program called Menin. And while at Pharmacyclics, BTK turned out to be a wild success, no question. But we had always been enamored with the idea of using biomarker strategy or techniques where you can really understand and identify the patient and the driving force behind their cancer. And then you can design a molecule that's meant for that specific driving force. And for BTK, you just enroll all patients with CLL. You don't know specifically if BTK is necessarily driving it at that one given point in time. And so we grew up, really enamored with Loxo and other companies that were going after these fusion mutations. And so when we learned that Menin was involved in these fusion mutations or gene alterations, we got very excited. And they had come to us with a reversible program. They had designed computationally many molecules, but synthesized maybe less than a handful, maybe 3 or 4 compounds. And the reason why they came to us is because they needed chemistry help. It took them about 18 months to make 3 molecules. And it's because, with computationally designed programs, the IP is wide open. The engagement to the target is extremely novel, but the chemistry is completely unknown. And so the synthetic schemes are unknown. And that can take a long time, especially if you are dependent on a CRO to try to figure out how to make these scaffolds. And so they came to us needing that chemistry help. I am a chemist by training. I started out at Gilead working on the Hep C cure program and also co-invented remdesivir. I didn't find out until we were into the pandemic in 2020, but that was a molecule that I helped design 10 years ago. And so all we did was just take a real pragmatic, logical approach to the design of reversible inhibitors to Menin. And as soon as we started executing the chemistry and the synthesis and finally being able to produce SAR, you need to make about 100 or 200 molecules to have a steadfast program. And so the rate of 3 compounds every 18 months isn't going to fly. And so you really had to go back, open up the chemistry toolbox and rework the synthetic schemes to be able to properly explore the scaffolds that they had designed. But the upside or the benefit of going computationally is that you don't do any compound library screening. You just throw that out the window. And the reason why you do that is because you end up with molecules that are very specific, highly selective to the target because they're designed from scratch. And that's the way you want to start. And the reason is because once you get your initial set of lead compounds, you spend the rest of the time optimizing for PK properties, not optimizing for selectivity, which is very difficult. And so as we kind of worked our way through, the reason why we were able to kind of close the gap is once we understood that reversible inhibition wasn't the most ideal way to shut down the pathway. We saw that there was a cysteine sticking out in the pocket. And I had just hired folks from Celera and Pharmacyclics, and we kind of looked at each other and laughed and thought, maybe this is the key. Maybe this is the way that we can really show ultimate control of the pathway. And sure enough, as soon as we started to build in warheads and redesign the scaffold a little bit to accommodate the architecture of the target Menin, it was off to the races. And it took us a few generations to get to 219, but as soon as you got to the molecule, you just knew. Once the data started flowing, it was very clear. And even though it did only take us 3 to 4 years, if we would have had the team already in place, it would have been much faster, to be honest. And so we're very excited about what 219 has in terms of a profile. And the promise of Menin is playing out incredibly well. And The reversible inhibitors in the clinic were able to show for the first time that single agent relapse refractory AML, you can get complete response. So that's very validating for the target. We think that can extend to other tumor types beyond acute leukemia. You just have to be able to control menin in a specific way. And then with 219 and with menin as a target in general, what we're learning is with the same molecule that controls menin in the setting of cancer, you get high cell killing capacity. In the setting of diabetes, you get controlled proliferation with the same molecule. I mean, imagine that. I've never seen that before. I've never seen a target that can control cell killing in one environment and cell proliferation in the other. And that just speaks to how incredible the target menin is.
Yeah. And that's fascinating. Tom, it's really great to hear, how you got here. I mean, for us personally as a team, and for me as an analyst, we look for companies that have actually sort of grown their projects, from the ground up, right? Because then you've actually had to work through a lot of problems that, when somebody in licenses, that also works, but that can lead to some other issues.
Mm-hmm.
Maybe you can just, help educate us a little bit on this thing called reversible and irreversible inhibition before we talk specifically about why you think in menin, an irreversible approach can be more potent. But walk us just through a little bit about the pros and cons of reversible versus irreversible inhibition before we talk specifically how that applies to menin.
Yeah, of course, it's very simple. You can think of two magnets, and reversible inhibition leverages weak magnets and covalent enforces very strong magnets that you can't pull apart. And at the end of the day, what you're trying to do is with a covalent inhibitor, think of like crazy glue, right? That once you form that covalent bond, that's it, it's permanent. So that's why we say irreversible covalent inhibition is you form the covalent bond and it can't be pried off, as if you have an incredibly strong magnet coming together with another one. And so the benefit there is that you have ultimate control of that target. It's no longer predicated or dictated by how much exposure you put in a patient from a plasma perspective. Once you form that covalent bond, it's over. And that becomes very attractive if you want to try to minimize exposure of foreign agents to a body, which of course you want to do. You don't want to create a system where you have to maintain exposure all day, every day, because what happens is eventually your body is not going to be happy. Your molecule is going to hit other receptors. It's going to hit other targets over time, and you're going to have tolerability and side effect issues. So we try to minimize that with the advent of irreversible covalent inhibition, because you only need a few hours of exposure. The body washes out the drug and you maintain effect. And that's really the key. That's really what we're trying to accomplish. Aspirin's a covalent inhibitor, or remdesivir is, by the way, a covalent inhibitor. 219 is a covalent inhibitor. All of our programs are irreversible covalent inhibitors that optimize control of the pathway and minimize exposure.
Yeah. And Tom, just to be clear, I think for our audience, when you say with reversible inhibitors, you need to have elevated sort of plasma levels because that drug doesn't bind covalently, meaning one and done to the pocket or something within that pocket, you need to keep the drug exposure, at higher levels, right? Consistently.
Every reversible molecule has a certain affinity towards a pocket or say two weak magnets coming together. And so all you're trying to do is ensure spatial orientation and presence of the magnet. And so reversible inhibitors have to maintain that exposure in order to occupy or control the pathway of interest.
And so that just leads us to, menin specifically, I guess, we've covered some companies with reversible inhibitors before, and it seems like there's some areas where maybe reversible inhibition could be advantageous, right? But it seems like in menin, Biomea's argument, and we agree with this, which is that irreversible inhibition is actually the way to go. So can you just talk specifically about what is it about menin that makes irreversibility, in your opinion, and in our opinion, a more potent approach than a kind of reversible approach?
Yeah, absolutely. And here again, it has to do with competition, right? It's all about competitive binding. That's what we're doing here, because your target's not designed for your molecule. Mother Nature designed it for something else. And so you're in constant competition. And menin is also in constant competition because it can form complexes with MOL as one option, but it can form protein complexes with other cofactors. And so, as you can imagine, you're trying to create a wedge to prevent menin from engaging with these proteins. And so the system or the body can use a multitude of proteins that have different levels of affinity. So as a reversible inhibitor, what concentration do I need? Well, it depends on what cofactor is responsible, and it depends on what's the affinity of the cofactor. And trying to predict that is very difficult. And that's why we thought early on, with even just, if you use just MOL, for example, MOL has over 100 different fusions. Those different fusion partners can fundamentally have different binding affinities. And so if you have over 100 different binding affinities, what concentration do you need to disrupt AF6 versus AF9 versus AF10? And we thought the FDA doesn't want you just to keep dosing up patients and then create an umbrella effect to cover all. They want you to really understand why you work in that patient, and really why you work in the pathway. And so we thought the advent of a covalent inhibitor, if we just dock on to menin and not worry about all the different binding affinities of all the different cofactors, that gives us great control and really achieve an optimal biological dose, not just for a handful of patients, but for many patients.
And then also just on the safety side, if you could just touch upon that, are you basically, because you only have to give it for, as you said, it's cleared after a few hours. Can you give lower doses or less dosing with your approach versus a reversible approach?
Yeah, the relative exposure just has to do with how potent you are in general. And then once you reach a certain threshold and you form the covalent bond to all the menin, right, and control the pathway, that's kind of key determinant factor. And just like you have specific assays to evaluate your reversible inhibitors from a selectivity specificity perspective, you have very similar assays for covalent inhibitors. So we don't go in blind, selecting a molecule like BMF-219 and hoping it's safe. You go through a very long list of assays to make sure that you selected the right molecule. You weed out covalent inhibitors that are promiscuous, that bind to any protein. It's a rigorous process. And so 219 rose up from the advent of many covalent inhibitors, not just a few. And it goes with the comfort of being a drug developer in covalent irreversible inhibition, is you just know the toolbox, right? Because this is where you live. And so with 219 was designed to be highly specific, highly selective. And so that safety perspective has to do not only with the architecture of the scaffold, right, the size, the shape, where else it can interact with, but also the position of the warhead that can also lend its selectivity index, so to speak.
Yeah, and just to be clear, Tom, when you're saying you did a lot of assays, like I guess one of the potential downsides of irreversible inhibition is that because it binds once and it's done, if there are any off-target effects, right, from a safety perspective, that could be deleterious, right? But what you're saying is that you went through a very rigorous process to first select a molecule that's very specific and selective for menin, and then secondly, you went through a ton of assays to make sure that there would be no sort of off-target effects to your approach with 219.
Yeah, that's right. That's exactly right. And at the end of the day, you pick proteins that don't have a weak turnover rate, right? You want proteins that have relatively short turnover for covalent inhibition, because then if there is a negative effect, you just stop drug. Very easy.
And it's kind of interesting because we want to maybe get back to this when we talk about your diabetes program a little bit further down here. Because the risk reward and what regulators and patients and physicians are willing to accept for cancer is much different, than for diabetes. But let's just talk about cancer first. If you can just talk to us a little bit about where exactly are you with your clinical trial right now? I believe you're in phase 1, right? In acute leukemias, and you might have actually widened the patient population, if I'm not mistaken, or thinking of doing it from acute leukemias. And then what are the steps you want to see with 219 in this initial area?
Yeah, that's right. At Pharmacyclics, what we learned is you want to go in parallel. You want to understand all the different tumor types at the same time, right? And what we did with 219 is we picked the tumor types that we thought would have the highest probability of success. The 7 tumor types is not the exhaustive list. It's just a list of tumor types we think 219 has the highest probability of showing great success. And so it's true, we're in our phase 1. We had FPI at the end of January. And the phase 1 is enrolling acute leukemia, and then also DLBCL and MM will happen soon. And KRAS activated tumors we anticipate by the end of the year.
Got it. So acute leukemia is DLBCL, and then KRAS accumulated tumors through this year is what the initial sets or target set you're looking at with 219.
That's right. Yeah. So we'll be exploring multiple tumor types. And again, we're not, it's not the approach of, look, we just hope it's going to work in a relapse refractory setting. We took our time from a translational perspective to really understand what are the subtypes within acute leukemia, within MM, within DLBCL that should have strong or high dependency on menin and therefore should work very well with 219.
And then, we have a question here, which is what are the benefits of and I think you kind of hinted at this with the Pharmacyclics approach. What are the benefits of focusing waiting for one tumor type to read out while you're also, are you going to wait for the others? You're kind of running them in parallel. So as one reads out, it also gives you some learnings into the other ones that you're sort of working on.
That's right. We run in parallel for sure, but we'll announce data as they come. So we won't wait.
Yeah. And then maybe you can just talk to us a little bit about how do you foresee, 219 sort of expanding, beyond its initial kind of set of targets. Will you kind of, it looks like it's multiple myeloma, DLBCL. Would you wait for the acute leukemia data to read out before you go there specifically, or would you just sort of, go ahead with recruiting patients for those indications?
No, we start as soon as possible.
Understood.
And at the end of the day, you want to do a dose escalation in each tumor type, so you understand the optimal dose for that particular tumor type. It's very important. And just real quick to take a little step back from a reversible, irreversible perspective, keep in mind that what we learned at Pharmacyclics was a patient can only benefit from drug while staying on drug. Right. And that's why we think irreversible covalent inhibition is so powerful, is because we think it lends you the profile for patients to stay on therapy for a long time, stay on that covalent inhibitor. That happened with ibrutinib, right? We have patients out to 4 or 5 years taking daily covalent inhibition of a BTK. And really it's because of that advent of the efficacy that you can achieve with just a few hours of exposure. And that's really critical to understand. It's why we picked, program 2, program 3, et cetera, is because of that notion. And so our intention is, we think acute leukemia patients can be helped if you keep them on drug. And the longer they stay on, the longer you can keep the disease at bay, the better the chance they have of improving their lives.
And just quickly, last question on 219, if you tackle this quickly so we can talk a little bit about diabetes, which is that you've got a one, maybe 2 folks potentially ahead of you in the menin space, right, in acute leukemias with the reversible. How do you see, your approach? I mean, do you think it's a zero-sum game? I've never seen a zero-sum game in the biotech space, right, in diseases. But how are you thinking of your approach versus other companies that are using a more, reversible approach and might be 1 or 2 stages ahead in clinical development?
Yeah, it's a great question. And so the zero-sum game is an interesting concept. And, because patients cycle through many therapies, it's typically not a zero-sum game. However, investors are correct that if you have a similar profile, it is 100% zero-sum game. You have to be differentiated. And if you have a profile that's in the lead and they get approved first, and then you have something that's very similar, even a year or 2 later, you're toast. And so with BMF 219, we think our profile is night and day. compared to what's already been shown in the clinic with the menin inhibitor. And so we're very excited to show the world what we think of 219.
Great, Tom. So Tom, maybe we can just spend a little bit of time on diabetes. You talked about how 219 kind of knocks down the proliferation of cells. Can you talk a little bit about the pathology of diabetes and what aspect of it could 219 address?
Yeah. So what's very interesting is, when you think about diabetes, right, this comes through the advent of beta cell exhaustion, right? And we learned about this early on in school that, type 1 diabetes immune system apparently is attacking your beta cells. And then in the type 2 setting, it's through the exhaustion of beta cells that they just kind of run out of gas. And the gas is this insulin, right, that's digesting your glucose. And so what we didn't learn in school is that, these beta cells are controlled from a proliferation perspective by menin, interestingly enough. So in the setting of current standard of care, you have these GLP-1s that essentially act like the fuel or the gasoline for beta cells. And essentially all they do is they squeeze more insulin out of each beta cell. And they do that quite well. With menin acts like the brakes, and it prevents beta cells from turning over. So if you could only turn over your exhausted beta cell and establish a new fresh pool of beta cells, you now finally are able to have homeostasis and glucose control, because now you have beta cells that can produce an adequate and functional insulin, which is key. And so all we're trying to do is use a menin inhibitor to basically turn over the pool of beta cells and then reestablish the ability to have insulin, normal, proper levels of insulin that can then process your glucose. And so what's so exciting for us is that this is not something where you have to continuously regrow a pool. You regrow the pool one time, and then you can observe. And perhaps there's maintenance down the road, but beta cells have a very long half-life, on the order of years, many years. And so what gets us really excited is that this could represent a real paradigm shift for these patients, because we would subject them for a short period of time with BMF 219, reestablish a new pool of beta cells, and observe.
Yep. No, that's fantastic. Is it just, will the therapeutic dose of 219 be different in diabetes versus oncology? I assume it will be, right?
Potentially slightly lower, but on par. And if you think about, look, there's certain hurdles, as you mentioned early on. There's certain hurdles that you have to achieve to get your molecule into the clinic for oncology. But again, it's a very different setting for cancer patients than it is for diabetes. And so you have to have a very clean molecule to get into diabetes. And what's amazing is, as we go through all the key toxicity studies for diabetes, 219 is passing with flying colors.
Yep.
Think about, wow, if you have that special of a molecule that can pass those toxicology studies, what an incredible drug this could be for oncology.
Yep. No, absolutely, Tom. It's fascinating for me. I've known you long enough to have seen you in various settings, and I've never seen you this passionate in my life. So that's great. It's great. It's really, really nice to see. I'll just end with diabetes. Diabetes. Just tell us exactly where you are in the preclinical stage and which division with the FDA you're interacting with. What are the steps you need to do in order to get into the clinic?
Yeah, so going through executing all the in vivo studies, we've been accepted to present at ADA. So expect us to present our in vivo data. We're very excited to have that opportunity, and basically now we're working through all the rest of the translational work that you need to do to support a successful IND filing. So we're doing that right now, and we're on track with our IND filing in the second half of this year.
Yep. Got it. And then maybe you can just talk to us a little bit about target number 2 and 3 that you might disclose 1 or 2 this year. How are you thinking about them, and what could they be, just generally speaking?
Yeah, generally speaking, look, as we increased our understanding of menin as a target, we wanted to select targets that would be complementary to menin. And the idea is that we want to have a suite of covalent inhibitors where we can build a fit-for-purpose proprietary combination based on the driving force of that patient and based on the tumor type. And so our goal is to execute program 2, program 3, and perhaps another program where we put together these proprietary combinations. Program 2 and program 3 can for sure stand on their own 2 legs as a target. And so, what we're doing is we're picking targets where folks didn't realize that covalent inhibition is possible or didn't realize the best design for a covalent inhibitor for this target.
Yep. Tom, we're at our time. I'm sure we could just keep on talking for a while.
Yeah.
I will hope to see you in the Bay Area in the next few weeks. You, Ramses, and the team are looking forward to keeping the conversation going.
Absolutely. Thank you so much.
Thanks, Tom. Thanks for taking the time.
Take care.